Hello, everyone, and welcome to today's session. And today we'll be discussing the theory related to the chapter Principle of Inheritance and Variation. So, in this session, we'll be talking about the NCERT section 4.2 and 4.3, okay, which is inheritance of one gene and inheritance of two genes. And this is the part 3 of the lecture. Before starting this lecture, I just want to give you one information that the images, text, and other materials used in our presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@iitk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So, what are the objectives of today's session? So, today we will be talking about codominance, which is again a deviation from Mendelian inheritance. Within this topic, we will first understand what is codominance. Okay, and how the inheritance occurs in the case of codominance and how it is different from the complete dominance. We will understand codominance using the ABO blood grouping. So we will take the example of ABO blood grouping system. So in this ABO blood grouping system, this is a very classic example of codominance. So we will understand the codominance, the mechanism of codominance with this example of ABO blood grouping. Then we'll talk about inheritance of two genes. Okay, within this, we'll be talking about the dihybrid cross. Okay, so we will understand the dihybrid inheritance pattern. So what is a dihybrid cross? Cross between individuals which differ in two traits or which differ in two genes. This is known as a dihybrid cross. And then we'll also calculate the dihybrid cross ratio. So here we'll be talking about the phenotypic and the genotypic ratios that we get in the dihybrid cross. Okay, so we'll be talking about inheritance of one gene, and within this topic, we'll be talking about codominance. We'll be talking about inheritance of two gene. Then we will summarize this entire lecture. We will discuss some NCERT-based questions which are based on these topics. And then in the end, we'll discuss some keynotes that will help you memorize these topics. And these keynotes will also help you crack the NEET exam. So here I will tell you which topics are important. Okay, and from which topics the questions generally come in the NEET exam. Okay, what should be your focus in the NEET exam? Now let's begin with today's lecture. So we'll be talking about codominance. Now, earlier we have discussed that the F1 generation shows two possible patterns. First is the complete dominance, where the F1 generation resembles one of the parent. So what happens in the codominance? What happens in the complete dominance? Okay, one gene or one factor or one allele, it masks the expression of the other allele. Okay, and in the F1 generation, only one of the parental phenotype is observed. Okay, the dominant trait is observed in the F1 generation. The phenotype of the dominant parent is observed in the F1 generation, and the recessive trait doesn't appear. So this is because of complete dominance. One gene is dominant over the other gene. Complete dominance can be, you can say, studied in heterozygous condition. For example, capital T and small t. Okay, these are the two alleles of the plant height. Capital T is dominant over small t. So here the phenotype will be tall. So this is because of complete dominance. Then In another case that we have studied, the F1, it shows intermediate phenotype. This happens in the case of incomplete dominance. Okay. Now there is a blending of traits here. And because of the blending of traits, the F1 shows intermediate phenotype, and this intermediate phenotype is different from the phenotypes of the parent. The classical example of incomplete dominance was snapdragon. Scientific name of snapdragon is Antirrhinum majus. Here, if the red color flower was crossed with white color flower, then in the F1, pink color flowers was obtained. This was because of incomplete dominance. What do you mean by incomplete dominance? Where neither of the allele is dominant over the other. Now let's talk about the codominance. Okay. So this is the third pattern which is shown in the F1 generation. First was complete dominance, second was incomplete dominance, third one is codominance. Now let's understand what is codominance. In codominance, the F1 generation resembles both the parents. In incomplete dominance, the F1 generation doesn't resemble any of the parent. In codominance, the F1 generation resembles both the parent. So now F1 generation shows three possible patterns: complete dominance, incomplete dominance, and codominance. Remember this. The question can come from here. What all patterns are possible in the F1 generation? The answer will be complete dominance, incomplete dominance, and codominance. Now let's understand what is the mechanism behind this codominance. So we'll take the example of ABO blood group. This is a classic example of codominance. Now, a good example of codominance is a different type of red blood cells that determine the ABO blood grouping in the humans. So there are different types of red blood cell. What does this mean? And these red blood cells, they determine the ABO blood grouping in the humans. Let's understand what does this mean. Let's first let's talk about the gene which controls the blood group system. Now, ABO blood groups are controlled by the gene I. This is a gene which controls the ABO blood groups. Now, let's understand about the plasma membrane and the sugar polymers which are present on the red blood cells. So, we have discussed that red blood cells, they are of different types. How they are of different types? Because of their plasma membrane. And there are some sugar polymers present on the plasma membrane of these red blood cells which make them different. So, the plasma membrane of the red blood cells, they contain sugar polymers protruding from its surface. So, the red blood cells, the plasma membrane of the red blood cells, they contain a sugar polymer. So, this sugar polymer is different. Okay. Now, the type of sugar is genetically determined. So, type of sugar which is produced, this is genetically determined, means it is controlled by the genes. So, here you can see, this is a figure showing you ABO antigen specificity. The ABO antigen differs by just one sugar at the antigen terminus. What does this mean? So here you can see these are different blood groups: A blood group, B blood group, AB blood group, and O blood group. This A blood group, the plasma membrane of the RBCs of the person having A blood group, this will have this antigen, which is shown here. And the person having B blood group, the RBCs of that individual will have different kind of sugar. So you can see here, there is a yellow sugar. Here, this is the blue sugar. Okay. In case of person having blood group A, this is an acetyl galactosamine. In case of person having B blood group, it is galactose. Then in the AB blood group, both sugars are present. And in O blood group, there is no sugar. Clear? So you can see one thing is common in all the blood groups. So these four circles you can see, these are common in all the blood groups. Only this sugar is different. This yellow color sugar, blue color sugar. these are different. That's why it is written the ABO antigen differs by just one sugar at the antigen terminus. So at the terminal position of this antigen having this 4 circle, this is known as the H antigen, and the difference is only in the sugar, the terminal. Okay. In case of A, it is yellow, this yellow color sugar, and this B, the sugar is shown as blue colored sugar, and in AB blood group, both these sugars are present. Clear? So these sugars are produced by the alleles of this I gene. So there are 3 alleles of this I gene: IA, IB, and small I. IA allele produces the IA antigen, IB allele produces the IB antigen, and small I, it doesn't produce any antigen. IA, IB, when both they are present together, then they produce both kind of sugar on the plasma membrane. Now let's talk about gene I and its alleles. So gene I has 3 alleles: IA, IB, and small I. So usually a gene have only 2 alleles, but here a single gene is having 3 alleles. Now, what are the function of these 3 alleles? IA and IB alleles, they produce slightly different form of the sugar. This I have already told you. IA produces a different kind of sugar, IB produces a different kind of sugar. Okay. Now, allele I does not produce any sugar. This we have also seen in the image. So here you can see the IA is producing a sugar which is labeled as yellow in color, and the IB is producing different kind of sugar which is blue in color. So IA is producing an acetyl galactosamine, IB is producing galactose. This blue color is galactose. IAB blood group, this consists of both the alleles. Okay, so both the sugars are present on the plasma membrane. This blood group I, blood group O, it consists this small I, and it does not produce any sugar. So there is no sugar present here. Clear? So this is the case of codominance here. When IA and IB, when these two both are together present, then both of them are expressed. Okay? When both of them are expressed, so this phenotype is different from the parents. Now, humans, the human cells, they are diploid. The animal cell, they are diploid. Okay? Diploid means they consist of 2 alleles. One allele they will inherit from father, another allele they will inherit from the mother. So humans, they are diploid organisms, meaning each person possesses 2 of 3 I genes allele. So there are 3 alleles for gene I, but humans, as they are diploid, so they will possess only 2 of these 3 alleles. So either they can have IA or IB, they can have IA or small I, or they can have IA and IA, they can have IB and IB. So there can be different combination of these 3 alleles, but they will possess only 2 of these 3 alleles. So there can be different combinations. We will see how many combinations exist. So this allows for multiple genotypes and the phenotypes. Now let's talk about allele dominance here. Now IA and IB are completely dominant over small I. So remember, IA allele is dominant over small I, and IB allele, this is also dominant over small I. Now when IA and small I, they are present, only IA is expressed because IA is dominant over small I. So here the blood group will be A. Now IB and small I, when they are present together, then IB is expressed because IB is dominant over small I, and here the blood group will be B. Now let's talk about codominance of IA and IB. Now, when both I and IB are present, then they are codominant. Means both the alleles are expressed in the F1. This is known as codominance. Now, both I and IB, they express their own sugar types. So, in the people having blood group AB, the RBCs will have both the kind of sugars on their plasma membrane. So, this result in red blood cells having both A and B types of sugar, and the blood group will be AB. This is an example of codominance. So, here you can see the blood group system, the ABO blood group system. It is demonstrating complete dominance also, because when IA and small I are present, IA will be expressed and blood group will be A. When IB and small I are present, the blood group will be B. So this is due to dominance. Law of dominance is being followed here. But in the case of IA and IB, codominance is being followed here. Okay. The question can come here that the ABO blood group system, it follows which laws? So it follows law of dominance. In this, you can explain the IA small I or and IB and small I. It follows codominance also. You can explain IA and IB when they are both present together, then both of them are expressed and the blood group is AB. It is an example of multiple alleles also, because this I gene, this has 3 alleles. So when more than 2 alleles are present, then this is known as multiple alleles. So this blood group system, this is also an example of multiple alleles. Now, since there are 3 different alleles of the gene I, there are 6 different combinations of these 3 alleles. Okay, and a total of 6 different genotype of the human ABO blood type. So let's have a look at this table 4.2, and you can calculate how many phenotypes are possible now. So this is a table showing you genetic basis of blood groups in human population. Now let's see here. In total, there will be 6 genotype and 4 phenotypes. So humans, they are diploid, so they will inherit one gene allele from one parent, another allele from the another parent. So these are the different combinations that can be made. IA. Here the genotype will be IA and IA. Blood group will be A blood group. Then another case, IA and IB. Here both the alleles are present, and this is a case of codominance, so blood group will be AB. Then IA and small I, genotype will be IA and I. Blood group will be A because of complete dominance. IB and A. This is again IA, AB. This IA and IB. This will be AB here. Then IB and IB. Genotype will be IB, and blood group will be B. IB and small I. This will be IBI. Blood group will be B because of complete dominance. And small I, when they are both present together, then the blood group will be O. And here no sugar will be produced. No sugar will be present on the plasma membrane. So here you can see there are 6 genotypes. How there are 6 genotypes? One is this one, second, third, okay, then fourth, fifth, and sixth. Because this one, this is AB and this is also AB. These two are same. So in total, there are 6 genotypes and there will be 4 phenotypes. How there are 4 phenotypes? Blood group A, okay, blood group AB, second one, then blood group B, third one, blood group O. So remember, there are 6 genotypes and there are 4 phenotypes. Okay, again question can come from here. How many genotypes are there and how many phenotypes are there? Okay, so this table is very important. Clear? Why there are 6 genotypes? Because this has been repeated twice. Now, what are the key rules of blood compatibility? Now, let's talk about ABO system rules and RH system rules. So, you know that the blood group can be A positive or B positive. Okay, A negative or B negative. So, what do you mean this? What do you mean by this negative and positive? So, there is one more antigen, RH antigen. Okay, isolated from rhesus monkey. So, that's why the name RH. Clear? So when this RH antigen is absent, then you write it as A negative. When Rh antigen is present, you write it as A positive or B positive or O positive, AB positive. So presence of Rh antigen, okay, when it is present, then you write plus positive. When the Rh antigen is absent, you write negative. Now, these are the basic rules of blood compatibility. Type A means blood group A. This has the antigen A, but it has anti-B antibodies. Okay. Blood group B, it has B antigens on its surface, but it has anti-A antibodies. Type AB has both A and B antigens, but it has no antibodies. And type O has no antigens, but it has both anti-A and anti-B bodies. Okay, clear? So blood group A will have anti-B bodies. If it has anti-A, if it has anti-A antibodies, it will kill itself. That's why anti-B bodies are present. Anti-B antibodies are present. And similar in the case of RH system rules, who can give blood to whom? So RH negative can give blood to both RH negative and RH positive. RH positive can only give blood to RH positive. RH negative patient can only receive from RH negative blood, and RH positive patient can receive both RH positive and RH negative. So these are the rules that should be followed during the blood transfusion. Okay, this is clear. So you can see this is very important to note here that RH negative can donate to both negative and positive. But RH negative patient can only receive from RH negative, because if it gets blood from RH positive, then RH positive has the antigen, and against this antigen, antibodies will be generated in RH negative person, and there will be coagulation of the blood. RH positive can receive from both RH positive and RH negative, because RH positive person will already have the antigen here. Clear? So now you can see in this table, now who can receive the blood and who can donate the blood. Okay, if your blood type is this, and from whom you can receive the blood. So AB positive person, okay, AB positive can receive blood from all of these. Okay, this is clear. Then AB negative can receive blood from these. Clear? Then AB positive person, who can receive, here it is written. Clear? So this is a table. You should remember this table, and you should follow all these rules. You should remember all these rules while transfusion of the blood. So O negative, this is the universal donor, not O positive. So this should also be noted. O negative is the universal donor. Okay, clear? So O negative can donate blood to all the individuals. So here you can see O negative. This can donate to all the individuals. Okay, see it this way. Yeah, so you should remember this table. Then AB, they are universal recipient. AB blood group. They can receive from all the individual. Now this blood group, this is also example of multiple alleles. So ABO blood grouping demonstrate multiple alleles. Now what are multiple alleles? More than two alternative forms of a gene. So when a gene have more than two alleles, this is known as multiple alleles. So if it has three or more alleles, this is known as multiple alleles. So blood group system is also example of multiple alleles. So three different alleles control blood type inheritance. Now, but individuals as they are diploid, they inherit only two alleles, one from each parent. Now, multiple alleles can also only be observed at the population level. So remember this, multiple alleles can only be observed at the population level, not at the individual level. Now let's talk about the pleiotropic gene control of starch synthesis and seed shape in the peas. Okay, so we'll discuss about pleiotropy. So there is a separate section in the book regarding pleiotropy, but here also we are discussing pleiotropy with reference to the starch synthesis and seed shape in the peas. So first of all, what is pleiotropy? When a single gene affects multiple traits, this is known as pleiotropy. So what happens usually? A single gene controls only single trait. Single gene controls only single character. But here you can see a single gene, if it controls or if it affects multiple traits, it is known as pleiotropy. Now let's understand this with the example. Clear? Now, gene B, suppose there is a gene B that controls starch synthesis and seed shape in the seeds. So these are two traits here, starch synthesis and seed shape. These are two traits. And gene B, single gene is controlling these two traits. That's why it is example of pleiotropy. Now first let's talk about starch grain size. So in the case of homozygous dominant, large starch grains are produced because there is quite efficient synthesis of the starch. Then in the case of homozygous recessive, small starch grains are produced. Then in the heterozygous condition, intermediate starch grains are produced. Okay, so first keep this in mind. So these are the phenotype that are shown to you, phenotype in the genotype. Remember this, in heterozygous condition, intermediate starch grains are produced. So this should be the example of what? We will discuss it. Now let's talk about the other character, seed shape. Capital B seeds are round. Now small b seeds are wrinkled. And in the heterozygous condition, capital small b, they also produce round seeds. So here there is no intermediate phenotype. So what does this demonstrate? So this demonstrates that the gene B in the case of starch grain size, okay, in the heterozygous condition, it is showing intermediate phenotype, means it is an example of incomplete dominance. Whenever intermediate phenotype is observed, it is example of incomplete dominance. In the case of seed shape, the same gene, this is acting as a dominant gene. Here capital B is dominant over small b. In the case of seed shape, because in heterozygous condition, round seeds are produced. In starch grains in size, here there is no complete dominance because intermediate starch grains are being produced. Okay, now you can see regarding seed shape, allele B shows complete dominance over allele small b. Okay, but regarding starch grain size, the alleles show incomplete dominance. So you can see this can also occur. A single gene is controlling two traits. Okay, this is known as pleiotropy. And the single gene can show dominance also, and a single gene, same gene, can show incomplete dominance also. So what conclusion do you make from this? Okay, so how you can say any gene is dominant or any gene is recessive? So how you will say by this? So you can see here what is happening. Let's see. Now the heterozygous phenotype is intermediate between the homozygous phenotype. This we have seen in the case of starch grain size. Now, so you can say dominance is not an inherent gene property. Now what does this mean? So this means that dominance is not an autonomous characteristic of the gene itself or its gene product. Dominance depends on multiple factors. First of all, it depends on the gene product itself, the product of the gene. Then the production of a specific phenotype from that gene product. And third one, the specific phenotype under examination, especially if the gene influence multiple phenotype. Okay, now let's understand this. So here, dominance depends on multiple factors. First factor is the gene product itself, the product of the gene. What is gene coding for? Clear? So the phenotype depends on the product of the gene, first of all. Then the dominance depends on production of specific phenotype from that gene product. What phenotype is being produced by that gene product? And then the specific phenotype under examination, specific character under examination. So like in the case we have seen of gene B, first there was starch grain size and another was the seed shape. In the case of starch grain size, when we examined the starch grain size, then the same gene was showing incomplete dominance. When we were examining the phenotype seed shape, then the same gene was showing complete dominance. So the dominance can depend on the specific phenotype or the specific character which is under examination. Okay, so you should remember this: a dominance of a gene depends on what? First, the gene product of the gene. Then that product leads to what phenotype? And then the type of phenotype that we are investigating, the type of character that we are investigating. So, what was the purpose of explaining this pleiotropy? Okay, pleiotropic gene control is starch synthesis seed shape in peas. So, the purpose of this experiment was to tell you, first of all, about the pleiotropy and about, okay, on what factors the dominance depend. Clear? So it depends on the gene product. It depends on the production of specific phenotype from that gene product and the specific character under the, or specific trait or specific phenotype under the examination. Because you can see a same gene is showing incomplete dominance when you are analyzing one character, and the same gene is showing complete dominance when you are analyzing another character. So dominance in gene isn't an inherent property, but depends on how genes interact with each other and their environmental conditions and the phenotype under the examination. This is the conclusion here. These are basically these 3 points. First point, second point, third point. Gene product itself, production of specific phenotype, and specific phenotype under examination. Now let's talk about the inheritance of two genes. So within this, we'll be talking about the dihybrid crosses. What are dihybrid crosses? Crosses between individuals which differ in two traits or two characters. So Mendel also performed crosses with pea plants differing in two characters. Okay, let's take an example of a dihybrid cross done by Mendel. So the pea plant with yellow color and round shape. So you can see here two characters are being analyzed here. Yellow color of the seed and the shape of the seed. These are two characters. So a pea plant with yellow color and round shape seed were crossed with a pea plant with green color and wrinkled shape. So these two plants that are being crossed here, they differ in two traits which are controlled by two genes. Okay, now what was the results of this cross done by Mendel? Now the gametes which were produced were capital R, capital Y and small y. And these unite on fertilization to produce the F1 hybrid, capital R, capital Y, small y. Okay, clear? So first you will, you can say, write the gametes which are produced by these two parents. Suppose yellow color is controlled by capital R, green color is controlled by small r. Round shape is controlled by, you can say yellow color is controlled by capital Y, green color is controlled by small y, round shape is controlled by capital R, wrinkled shape is controlled by small r. Okay, clear? And these will be homozygous, you can say pure breeding lines. So they will be homozygous for a particular trait. So Mendel, he always crossed pure breeding lines or true breeding lines, which were made by self-pollination. They were made by repeated selfing. So these are homozygous for the traits. So one parent will be homozygous dominant, another parent will be homozygous recessive. So genotype of one of the parent will be capital R, capital R, capital Y, and the genotype of other parent will be small r, and small y, small y. So the gametes produced by this parent, yellow color and round shape, this will be capital R, small y, and the gametes produced by green color and wrinkled shape seeds will be small r and small y. Now you can make a Punnett square and then you can make all the possible genotypes. So in the F1, all the plants will be having the genotype capital R, small r, capital Y, small y, and the phenotype will be round and yellow seeds because of the law of dominance. Okay, so these plants displayed round shape and yellow colored seeds. So in the F1 generation, round shape and yellow colored seeds were observed. Now, what were the dominant traits here? So in the F1 generation, only dominant traits are observed. Okay, so what will be the dominant traits? We can say yellow color was dominant over green color because yellow colored was seen in the F1 generation. And round shape was dominant over the wrinkled because round shape was observed in the F1 generation. So by looking at the F1 generation, you can tell which character is dominant and which character is recessive. Now, what are the alleles of the seed trait? So here we have seen now for the seed color, capital Y for yellow and small y for green. For seed shape, capital R was for round and small r was for wrinkled. And parental genotype was capital R, capital Y, and small r, small y. So these were the parental genotype. These were crossed with each other. So gametes produced by the parent having capital R, capital Y, capital RY will be capital R and capital Y, and the gametes produced by the parent having small r, small y, small y. The gametes produced will be small r and small y. Then you can cross, and after fertilization, the F1 will have capital R, small r, capital Y, small y. So you can see in this image, this is showing you a dihybrid cross. This is showing you the inheritance of two genes. So this is the P generation, means parental generation. So the plant having round and yellow color seeds were crossed with a plant having wrinkled and green color seeds. The genotype is also written here. So these are pure breeding lines. So they are homozygous for the traits. Then the gamete, you will write the gametes produced. So you can see the gametes produced by this parent. Homozygous dominant parent will be capital R, small y. Gametes produced by homozygous recessive parent will be small r, small y. Then in the F1 will be of round and yellow color because round color is dominant over round shape is dominant over wrinkled and yellow color is dominant over green. Genotype will be capital R, small r, capital Y, small y. Then after selfing of the F1 generation, you will get the F2 generation. Then again, you can write the gametes of male on one side and female on other sides, and you can make all the possible combinations here. And then what phenotypic ratio you will get here? 9:3:3:1. This is the phenotypic ratio you will get here. In the case of monohybrid growth, you used to get 3:1 ratio. But here the phenotypic ratio is 9:3:3:1. Okay, this is clear. So this is a dihybrid cross. This is showing you inheritance of two genes. Now, Mendel observed that the dihybrid cross, the results of the dihybrid cross. So we have seen the phenotypic ratio of 9:3:3:1. So this dihybrid cross, this was consistent with the monohybrid cross. Now what does this mean? So what Mendel did? So Mendel, he first self-hybridized the F1 plants, and then he found that the F2 generation split into 3:1 ratio. Okay. Now we were studying the dihybrid cross. So Mendel, what he did, he said that this dihybrid cross, the results of the dihybrid cross, these are consistent with the monohybrid cross. So what he did? He did separate monohybrid cross. Okay. Then what he obtained in separate monohybrid cross means, what do you mean by separate monohybrid cross? He crossed the plant having yellow color seeds with green color seeds, and then separately he crossed the plants having round shaped seeds with wrinkle shaped seeds. And what he observed? He observed the typical 3:1 ratio in the F2 generation for both the cases. So in the F2 generation, split into 3:1 ratio. So yellow-green split in 3 dominant and 1 recessive ratio, and round and wrinkled also segregated in 3 dominant and 1 recessive. So these were the ratio that we used to get in the monohybrid cross. Okay, so Mendel conducted his separate monohybrid crosses. Now what Mendel did? Now these results match the typical monohybrid cross patterns. Okay, now what Mendel did after this? So Mendel said, Mendel crossed these two. Okay, so Mendel crossed this 3:1 ratio with 3:1, and he got the 9:3:3:1 ratio. Okay, so what does this conclude? This concludes that the dihybrid cross, these are the result of separate monohybrid cross. Okay, so yellow and green color, that was showing 3:1, and round and wrinkle, that was showing 3:1. So when you merge these two, you will get 9:3:3:1 ratio. So by this, you can conclude that the dihybrid cross, these are nothing but they are the combinations of two monohybrid crosses. Okay, now let's summarize this entire lecture. So first of all, we talked about the codominance and the inheritance of two genes here. So what was codominance? In codominance, both the alleles are expressed equally in the F1 generation. Okay, then both the alleles are expressed equally, neither is dominant or recessive. Both the traits appear together in the heterozygous condition. Okay, now you can see here, single gene has multiple alleles. The example is ABO blood group. This is a classical example of codominance. Okay, you can study codominance in single gene crosses, means in monohybrid crosses. That was codominance. That's why codominance is kept under inheritance of one gene. Now, two gene inheritance involve inheritance of two genes controlling two different traits. Now here each gene segregate independently into the gametes. So we'll talk about this when we'll talk about law of independent assortment. Here each gene segregate independently into the gametes. Now next, create more complex phenotypic ratio compared to single gene inheritance. So in the monohybrid cross, 3:1 ratio was observed, phenotypic ratio. In dihybrid cross, 9:3:3:1 ratio is observed. So this is a more complex phenotypic ratios compared to monohybrid crosses. You can study two gene inheritance by dihybrid crosses. Example is seed color and seed shape in peas, where you get 9:3:3:1 ratio. Now let's discuss this question. In a pea plant, yellow capital Y allele is dominant over green small y, and the round capital R is dominant over the wrinkled small r. What is the phenotypic ratio when crossing plants that are both heterozygous for both the traits? Okay, you have to calculate the phenotypic ratio. Okay, when both the heterozygous parents are crossed with each other. What would be the answer to this question? Now let's calculate the phenotypic ratio when crossing plants that are both heterozygous for both the traits. Okay, so these are the parents given to you. The genotype of the parents are given to you. Okay. So first of all, this is the cross between two heterozygous individuals where capital Y is dominant allele Small y is recessive allele for green color. Capital R is dominant allele for round shape. Small r recessive allele for the wrinkled shape. Now, what are the gametes produced? So each parent can produce 4 types of gametes here. So these are the 4 gametes that will be produced by each parent here. Now you can make the Punnett square. So this will be the Punnett square. You can make all the possible combinations using the Punnett square. And then you can calculate the phenotypic ratio here. So what is the phenotypic outcomes here? So yellow round will be 9. So total there will be 16 genotypes here. Okay, there you will get 16 possible genotypes here. That's why we are writing 16 here. So yellow round will be 9. So there will be 9 by 16. Yellow wrinkle will be 3 by 16. Green round will be 3 by 16 and green and wrinkled will be 1 by 16. So this is 9:3:3:1 ratio. Okay, so the phenotypic ratio in dihybrid crosses, these are 9:3:3:1. Okay, so you get this ratio when the genes show complete dominance and when they assort independently. Next, a man with blood group type AB has a child with a woman with blood type O. What are the possible blood types of their children? You have to show your solution using a Punnett square. So what will be the answer here? What will the possible blood types of the children's when the parents' blood group is AB and the mother's blood group is O? What will be the answer to this question? And the possible blood type of the children will be, so parent 1. Okay, the blood group of parent 1 is AB, so it has both the alleles, IA and IB. Parent 2 has O blood group, so it has small I alleles. Now you know there are 3 alleles for gene I, IA, IB, and small I. Now we can make the Punnett square here. So you can see, okay, you will write the alleles of parent 1 here or gametes of parent 1 here. Okay, these are the alleles, IA and IB. This is of parent 1 and these are the 2 alleles of the other parent. Now you can make the Punnett square and you can make the different, you will get all the possible genotypes here. Now what are the possible blood type of the children? So IA and small I, this will give blood group A. IB and small I, this will give blood group B. So each child has 50% chance of being of blood type A and 50% chance of being blood type B. So this IA and I, this will be A blood group, this will be A blood group. This one will be B blood group and this one will be B blood group. So 50% of the children can have blood group A and 50% of children will have blood group B. The next question, a plant with red flowers is crossed with a plant with yellow flowers. Genotype is also given to you. In the F1 generation, the flowers which are produced have both red and yellow patches. This is an example of what? So now you can see in the F1 generation, in the hybrid, the flowers have both red and yellow patches. So this is the example of what? Both the alleles, both the genes are being expressed in the F1. This example of what? Is example of codominance, incomplete dominance, codominance, or multiple alleles. So answer is this example of codominance. What is the explanation here? When the flowers show distinct patches of both parental colors, red and yellow, rather than a blended color, which can be orange. So if it is orange color, then it was incomplete dominance, example of incomplete dominance. But here, both red and yellow color patches was observed in the flower. So this is example of codominance because both alleles are being expressed independently and fully in the heterozygous condition. Now let's discuss this keynote. This will help you in your exam as well. So we talked about the codominance where both alleles are expressed equally in the heterozygous condition. You have to write this. They are expressed both in the heterozygous condition. There is no blending of traits here as seen in incomplete dominance, and there is equal expression of both the alleles. Then you can see example is ABO blood group system. And what is important regarding NEET? So you can get question of ABO blood group and there can be question on the phenotypic ratio calculations. And we have also discussed this type of question here in this session. Then regarding multiple alleles, so what is the definition? 3 or more alleles of the same gene. Okay, clear. But as humans, as the animals, they are diploid, they can have only 2 alleles. Yeah, even if a gene has more than 2 alleles, but the individual will have only 2 alleles. So there can be different genotypes and phenotypic combination. Example is ABO blood group system, where there are 3 alleles of I gene, IA, IB, and small I. Okay, NEET focus will be blood group inheritance pattern and genotype phenotype relationship. Okay, so like we were talking about in the ABO blood group system, there are 6 genotypes and there are 4 phenotypes. Now we talked about inheritance of 2 genes also. Here, this is very important. Here, independent assortment of gene occurs. We will discuss this in detail in the next session. What is the phenotypic ratio here? 9:3:3:1. You get this phenotypic ratio in the F2 generation. We'll talk about the genotypic ratio also in the F2 when we in the next session when we'll be talking about inheritance of 2 genes. Now, you get 16 possible phenotypes, 16 possible genotypes. Okay, in the F2 generation. Here there is independent assortment of the genes. Okay, and you can get questions on the probability calculations, like what is the probability of they will give you a particular genotype, so they will ask you like what is the probability of this genotype to occur, 25% or whatever, 50%, or how much, 9/16 or 3/16. What is the NEET focus here? So they can give you dihybrid cross problems, and they can give questions or phenotypic ratio or genotypic ratio calculations. All right. So with this, we have discussed the theory. Now let's discuss the DPP related to these topics. So let's begin with this question now. According to Mendel's dihybrid cross result, what phenotypic ratio is observed in the F2 generation? This is very simple question. So they are saying that in the Mendel dihybrid cross means a cross involving, okay, or what is dihybrid cross? When the crosses are made between individuals that differ in 2 traits. What is the phenotypic ratio observed in the F2 generation? But options here 3:1, 9:3:3:1, 1:2:1, and 1:1:1:1. What would be the correct answer here? Correct answer here will be option number B, 9:3:3:1. This is the phenotypic ratio in the F2 generation of a dihybrid cross, which shows complete dominance and independent assortment of the gene. 3:1 ratio is the phenotypic ratio in the monohybrid cross. 1:2:1 is the genotypic ratio in the monohybrid cross. Then 1:1:1:1, this is the test ratio in the dihybrid cross. Next question, which law of Mendel is illustrated by the independent segregation of 2 pairs of genes during the gamete formation? Okay. So when the genes, they assort independently, or when there is independent segregation of 2 pair of genes. So in the dihybrid cross, there were 2 genes that were controlling 2 different traits. And these 2, you can see these 2 genes, they were assorting independently. There was independent segregation of the 2 pair of genes. That too, this occurred during the gamete formation. So what law does it indicate? Law of dominance, law of segregation, law of independent assortment, of law of linkage. Correct answer here will be law of independent assortment. Okay, we'll talk about this in detail in the next session as well. What is law of independent assortment? Now, in a dihybrid cross between capital R, capital Y, and smaller parent, what is the genotype of the F1 generation? So first of all, what you will do, you will make the gametes of each of the parents, and then you can make the Punnett square, and then you can calculate the possible genotypes of the F1 generation. So these are the 4 options. What would be the correct answer? See, what you will do first, you will make the gametes of one of the parents. So one of the parent is homozygous dominant, another parent is homozygous recessive for both the traits. So gametes produced by capital R, capital Y will be capital R, capital Y. Gametes produced by small r, small y will be small r, small y. So the F1 will be capital R, capital Y, small y. So answer will be option number A here. Okay, clear? Now, in Morgan's dihybrid cross experiment, the deviation from Mendel's 9:3:3:1 ratio was due to. Okay, we'll talk about this also. So Morgan, he conducted experiments on Drosophila. Okay, he conducted dihybrid crosses in the Drosophila. And what he observed, he didn't observe this ratio 9:3:3:1. He observed some other ratio. Okay, so why was this deviation from this ratio 9:3:3:1? So what he observed that the genes were very, very closely, you can say, associated to each other. So they were physically associated to each other and they were present on the same chromosome. And the distance between the two genes was very less. So they were inherited together. So these genes were inherited together because the distance between these two genes was very less. And they were present on the same chromosome. So why there was deviation from this 9:3:3:1 ratio? Was it due to incomplete dominance, codominance, linkage of gene, or multiple alleles? What will be the correct answer here? Okay, the correct answer was linkage of genes. So genes were linked together. Okay, so when the genes are linked together, they are inherited together, and the typical ratio 9:3:3:1 is not observed. Now next, in the Mendel dihybrid cross, the F2 phenotypic ratio of 9:3:3:1 is obtained because, so typically in the F2 generation in the dihybrid cross, 9:3:3:1 ratio is observed. Why this ratio is observed? Because the two genes are linked and do not assort independently. Next, two genes are on the same chromosome but far apart. Next, two genes are on different chromosome and assort independently. And next one gene is dominant and the other is recessive. What will be the correct answer here? Okay, the correct answer here will be option number C. The two genes are on different chromosome and assort independently. Then 9:3:3:1 ratio is observed. Okay, when there is linkage, then there is a deviation from this ratio. So when the genes, they assort independently, and when they are present on different chromosomes, then you get this phenotypic ratio 9:3:3:1. Okay, we'll talk about law of independent assortment in detail in the next session. Okay, clear? So this independent assortment means one pair of allele assort independently or segregate independently of the other. Segregation of one pair of the allele Okay. It happens independently of the other pair of alleles. So we'll discuss this as well. So here the answer is option number C now. Okay. So with this, we have discussed the DPP also. Okay. We'll end this session now. I hope the session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening. And you can see all our answers are correct. Okay. Thank you very much for listening.